One second. Let me introduce you to Professor Chevrollier. He has his PhD in molecular and cellular biology. Let me also say before I finish the introductions is this is the first of what we hope to be many presentations sponsored by CYTOO. CYTOO is a company that makes plates that have micropatterns for cells to attach in either individual cell or multi-cell. And we hope to be able to share people's research who are working with CYTOO micropatterns to show you the vast different things that can be done with micropatterns. So our first one is we have a pleasure to present Dr. Chevrollier. He has a PhD in molecular and cellular biology and biochemistry. He studies mitochondrial bioenergetics and dynamics at the University of Nantes in Angers, France. And the picture we have here, the left-hand top picture, is actually the picture of the research of his research facility. Okay. The research team studies neuropathology and mitochondrial bioenergetics and dynamics. It's with the Department of Integrated Neurovascular and Mitochondrial Biology, and the folks that are involved are Danielle Henrion, Pascal Ranier, Vincent Procaccio. I hope I said all of that correctly.
Okay, yes.
Very good. So the research topic today is on neurological and neuromuscular pathology, talking about diseases associated with optic atrophy. So this is associated with mutations in mitochondrial genome. They're responsible for, just one second, I have a thing that's blocking me from seeing everything, so just 1 minute here. Okay. Responsible for mitochondrial defects in numerous patients. The clinical manifestations of mitochondrial DNA-related diseases may be variable even within the same family. So Dr. Chevrollier set up a biobank of fibroblasts to serve as models for investigating bioenergetics in the context of diseases due to defective mitochondrial dynamics. So they obtained skin fibroblasts from patients carrying OPA1, MFN2, GDAP1, and MT and mitochondrial DNA mutations, as well as from controls. The cells allowed the characterization of molecular and biochemical processes involved in mitochondrial dynamics of disease. And so now I'm going to turn over the presentation to Dr. Chevrollier so he can actually more thoroughly discuss his work. So I think you're in control now.
Okay, great. Okay, it works?
Yeah, it works.
Yes, so hi everybody. Thanks for the opportunity to present our works on mitochondrial analysis standardization by using CYTOO chips. So a mitochondria is are involved in fact in energy metabolism. I've got a go-to-meeting control panel here. It's just okay. So mitochondria are involved in energetic metabolism, so to produce ATP, but also in oxidative stress by the production of oxygen species, and of course mitochondria are involved in apoptosis process. Mitochondrial disease represent a broad spectrum of clinical and disorder reflecting the reliance of organ system on oxidative phosphorylation. So mitochondria produce ATP for brain, for heart and liver, and when there is problem with this ATP productions, most of the time there is trouble with the eye and sometimes a neurodegenerescence of the optic nerve leading to optic atrophy. Actually, in fact, more than 100 genes are directly associated with mitochondrial disease. NTD-encoded or nuclear gene. But in fact, it's probably just the tip of the iceberg. Mitochondria have probably a role in rare disease such as cancer and also cancer, stress and metabolic disorder, neurodegenerative disease, Parkinson, Alzheimer, and probably aging too. So the first image, in fact, of mitochondria were the electronic microscopy ultrastructure image like this, with the external membrane, the inner membrane, the cristae. But in fact, we know now that it's just a small part of a huge mitochondrial network, as you can see here in blue. Mitochondria form tubular structure with different size and shape. So there is a new world, a new function for mitochondria, which is the dynamics of mitochondria. And mitochondria have this type of shape, just small tubules, isolated tubules. The mitochondria could have this donut form, or could be connected with different branches. There is dynamics of mitochondria is also means that there is a migration of mitochondria through the cytoplasm towards the energetic need location. Few genes, few proteins have been involved, in fact, in this process, such as MFN2, which is a protein of the external membrane. This protein anchor and the close mitochondria and improve the fusion of two mitochondria. OPA1 is also a protein of the inner membrane involved in this fusion process, and also involved in the remodeling of the cristae structure. So there is an equilibrium between fusion and fission processes. In fact, MFN2, GDP1, and OPA1, well, to say that just MFN2 and GDP1 have been involved in Charcot-Marie-Tooth 2A type and 4A type, so with neurodegenerative disease. OPA1 is involved, OPA1 mutation lead to dominant optic atrophy. So there is direct link between protein involved in dynamics and neurodegenerative disease. So actually, to study mitochondria in pathological condition, so following mutation, or to evaluate chemical consequences, we apply a different study for the bioenergetic function of mitochondria, such as oxygen consumption, biochemistry techniques, and such as enzymology, or an approach of molecular sequencing, such as to sequence the mtDNA. So now we think that it's essential, in fact, to study the structure and the dynamics of mitochondria. For that, we use skin fibroblasts from patient and control. But the limit in classic cover slip was that the cells spread with on the glass slide with different extension, and the size of the cells are very different. We could have also some overlap. So we use CYTOO chips and the pattern in Y, in fact, to standardize the shape and the size of the cell. And we designed 3 micropatterns named L, XL, or XXL, in fact, to stretch in maximum the cell. In fact, our objective was to limit the thickness of the cells and to limit the overlay of the mitochondrial tubules. So our protocol is quite simple. In fact, we spread 6,000 cells per well in a 6-well plate. Here there is the CYTOO chip. 4 hours in DMEM medium with 1% of serum. After that, we load with mitochondrial 15 minutes. And after we mounted this CYTOO chip in the CYTOO chip chamber in DMEM without red phenol. For the acquisition, we use a fluorescence video microscope like this with a 1x oil objective mounted on a piezoelectric system. In fact, this so this is a microscope here, and we scan in z-axis the different plane, around 20, to keep all the information of the fluorescence signal. We apply a deconvolution algorithm named Huygens software, and after that we are using Imaris software. So first of all, to resume, we check the quality of the triangular cells like this in fast contrast. We need to show correctly the nuclear border. We scan, in fact, the cell like this. So we keep all these Z-plane here to obtain all the signal. We apply the deconvolution and we apply the 3D skeleton by Imaris. By this way, we are able to know the connected and unconnected part and to have a precise quantification of each filament. So here is the small movie to resume. So here we apply also convex Hull algorithm to calculate the volume of the cell. This is very useful, in fact, to realize the quantification of the mitochondrial volume per cellular volume. Yeah, another time. Fluorescence, mitochondrial network. So it's a different part here in yellow or blue of the connected mitochondrial network. And the green spot is the tip of the mitochondria. Okay, so here if it's an overlay on the Y pattern. There is also another possibility for a quick overview. In fact, it's surfacing, but it's less efficient for the quantification. And with filament tracer, it's better, in fact, to know the length of each tubules and the number of connection, number of branches. For mitochondrial network fragmentation, we use uncoupling molecules such as CCCP, and it's we have a well fragmentation of the mitochondrial network. So we apply this approach on a fibroblast from patient affected by CMT2A, so Charcot-Marie-Tooth 2A type, and with a MFN2 mutation. And this lead to increase mitochondrial connection, and the number of branching is increased. With the opposite, in fact, with OPA1, there is a fragmentation of the mitochondrial network. This fragmentation is sometimes associated with a decrease in the mitochondrial volume per cell, as you can see here. But with the mutation here, with this mutation, there is an increase in mitochondrial unconnected tubules. By this approach, what is very interesting is that there is different step, in fact, in the fragmentation of the mitochondrial network, and here is it could be the maximum, in fact, of the fragmentation. In this case, we have very small mitochondria with isolated mitochondria. And here in this, the only part of it, the only connected structure emerged from one point. This reticular aspect of mitochondria emerged from the microtubule organizing center. It was preserved despite the fragmentation of other mitochondrial regions. This result underscores the importance of OPA1 in mitochondrial biogenesis and in cellular mitochondrial organization. So this point is very important in probably in the organization of the cell. So that's why we CYTOO chips probably offer a new mean, in fact, to understand the different subpopulation of mitochondria within the cell. There is probably a perinuclear mitochondria here in blue, so because there is only just one tubules around the nucleus. The other part is the periplasmic mitochondria with the longest mitochondrial tubules reaching the corner of these cells. And here, probably the most important mitochondria are these central mitochondria emerging from the microtubule organizing center and connected to the endoplasmic reticulum and Golgi apparatus. So this conclusion, in fact, is that this CYTOO and this standardization of the mitochondrial network is very useful to first describes and quantifies the skin fibroblast from patient, but it's also very interesting to study the fission processes and to study mitochondrial biogenesis or mitophagy in different conditions such as energetic metabolism, affect of apoptosis, or different pathological condition or neuropathy. So thanks for your attention. So of course, if you have an amazing cell line, I'm very interesting. So thanks. If you have any question.
So yeah, we can take any questions. You guys I've unmuted everybody from my end, and if you have questions, feel free to answer or to ask.
Can you hear me?
Yes.
Hi, this is Giovanni Manfredi in New York. I wanted to know if for neurons, if this has been tested for, if there are any specific plates or format that would work for primary neurons.
For looking at mitochondria or for?
Yeah, mitochondria, yes. Exactly for, well, similar to what has been presented.
Pauline, do you know of any mitochondria with neurons? I don't. Do you?
No, I never tested.
No, either.
No. It would be an interesting. It would be an interesting thing to try. There's CYTOO plates. We do have CYTOO plates that can be used for neurons, but yeah. I don't know whether nobody's tried looking at mitochondria in that.
So what would be the shape of the wells or the grooves basically on those?
I think that we would have to explore it. So we have a neuro plate that is in development, and essentially we have multiple different shapes in that depending upon what the goal is. So some of the stuff is to look at neural toxicity. So with neural networks essentially, where the different cells can attach, but that's still being developed. But So one of the ways that we do work is we do work with clients to help develop what the shapes and stuff should be, and we're able to do custom patterns so that you can explore with our help, you know, what would be the best shape for that. I can share some of the neural stuff with you if you would like me to send you an email on just what we've done.
Yes,
Okay.
absolutely. Yeah, we'd like to look at it.
Where are you located?
At Cornell in medical school.
Okay. At the Cornell Wheel School?
Yes.
Yeah, we were just there. Not at Cornell. We were at Rockefeller a couple weeks ago.
Across the street from, yeah.
Yeah. Okay. So that's fine. I'll be glad to send that to you.
Also, I had another question. I mean, like, we have a Leica confocal. It's a different model, but is there any reason that to use the particular instrument that was shown in the presentation, or any confocal would do, as far as you know?
Yeah, probably. In fact, the limit is really for the IMRS 3D reconstruction, where you really need a high signal, and you need to have a good ratio signal on background. So probably with a confocal it's also perfect. So, but it's our tool, in fact.
Our stronger lens is 63x. We don't have 100x, but it should work anyways, right?
Yes, it should work. Yes, with a good, now there's no real difference between 63 and 100.
Dr. Chevrollier, can you hear me?
Yes.
I had a question regarding the pixel resolution that you achieved for the images, because you know, since you're doing this in computational analysis, it's really the size of the pixel that's going to matter, right?
You would like some detail about the pixel size of the image?
Yeah, I was just curious. Yes, because I'm curious if I could achieve that with our 60x objective as well.
It's 64.5 nanometers per pixel, in fact.
Well, that's very good resolution.
Yeah.
Okay. And a follow-up question to that regarding the mitotracker green. Now, are you concerned that the mitochondria, when they depolarize, that you lose mitotracker green staining?
Yes, we currently use mitotracker because it's, in fact, in this condition, it provides the best signal as compared to plasmid or to transfection system. That's
okay.
That's all.
Great.
Yes, and there's no toxicity in this time, the current time, so it's okay.
Oh, great, thank you.
Are there any other questions? So if there are no more questions, let me finish one thing. If you'd like me to follow up with you, or you guys all got emails from Brian, let him know, and I'll be glad to follow up with you on to talk to you about perhaps your application and maybe what we can do to help with that. And also, if you need more information on the micropatterns and a little more basic information, I also would be glad to share that with you. So if there are no more questions, I'd like to really thank Professor Chevrollier. That was very interesting, and I think it's a good addition for our very first presentation.
Okay, thanks.
So thank you very much. And thank you everybody for attending.
Thank you.
Bye.
Thank you. Bye-bye.